In this video, I will answer the question, how can we detect the mutation using the sequencing method? And we'll be talking exactly about DNA sequencing method discovered by Frederick Sanger and published in 1977. This is so-called the chain termination sequencing method, and it is used in diagnostic in order to verify if the mutation is present in the particular part of the genetic material. In this method, the special type of nucleotides are used. They are called dideoxynucleotides. And what is special in these nucleotides? They are dideoxy, so it means that they don't have the OH group at 2' end as every deoxynucleotide, but also they don't have the OH group at 3' end. So it's one of the modifications. And apart from this, these dideoxynucleotides, they are marked by fluorescent dye. So we have cytosine nucleotide marked with particular dye, thymine nucleotide marked with particular dye, adenine nucleotide, and guanine nucleotide. So not only they are lacking the OH group at 3' end, but additionally they have attached the colorful marker. And in order to sequence particular area of the genome, first this particular area of the genome is amplified by PCR reaction. And once we have the amplified fragment by PCR reaction, as presented here, there is another reaction similar to PCR, and the goal of this second reaction is to prepare the DNA strand to be sequenced. So in this reaction, we are using only one starter, forward or reverse, or we can use both in parallel. And what's going on? Starter binds to the template, and then we have the mixture of nucleotides. We have classical standard deoxyribonucleotides, and in addition we have these strange modified ones which do not have the OH group at 3' end, and additionally they have the colorful marker. And reaction similar to PCR starts, but only with one primer. So, and the nucleotides are added randomly, but every time when the colorful one is added, termination takes place, because after colorful one, we cannot add the subsequent nucleotide, since the colorful one does not have the OH group at 3' end, and we know that OH group at 3' end is crucial to add following nucleotide. So after this reaction, it's statistically confirmed, we have all possible fragments with all possible lengths terminated by colorful nucleotide. On the slide one more time, you see why after attachment of colorful modified nucleotide we cannot add the next one, because the lack of the OH group at 3' end prevents adding the subsequent nucleotide. So if we have the mixture of fragments terminated by colorful nucleotides, now we can organize them from the shortest to the longest using special type of electrophoresis. And during the special type of electrophoresis, these fragments are migrating, and the fluorescence is red. First we have the laser which excites fluorescence, and then we have the reader which reads the color. And if these fragments are traveling, migrating during electrophoretic field, we can read the signal, and every color is particular nucleotide. And we start from the shortest, and subsequently the longest are appearing. They are simply in order. So this is how the method looks like. And now I have for you several questions, so you can confirm if indeed you understand the Sanger sequencing method. So on the slide we have the questions, and in a moment we will answer all of them. So what happens if the concentration of labeled dideoxynucleotides is inappropriately selected, for example, too high or too low? We start with too high concentration, and if we have too high concentration, like presented in this part, And there is high chance that colorful nucleotide will be immediately incorporated when the reaction will start. So preferentially, the short fragments will be formed. And as a consequence, we can read finally only the beginning of the template, because we can only read this part. So if we have the product form on the template, preferentially we can read this part of our DNA dedicated to be sequenced. If the concentration is too low, like it's presented here, there is the low chance the colorful nucleotide will be incorporated at the beginning because the concentration is low. So preferentially, the long fragments will be formed. And as a consequence, we can read the end of our sequence, like we have here. So only based on this part, the sequence can be read, and only this area, for instance, we can read. What picture will we get when the patient is heterozygous in terms of mutation being analyzed? So here we have some area of the DNA between exon and intron. You can see exon, part of the exon and part of the intron. In the control DNA, in this particular position we are interested in, we have the guanine. If the patient is homozygote and the patient has mutation, we see signal from thymine in this particular position, whereas if the patient is homozygote and he has two alleles, one with guanine, one with thymine, we can see two signals in this position, because we have the signal from guanine and the signal from thymine, so two colors at the particular position and two peaks can be observed. What picture will we obtain if the patient is heterozygote in terms of insertion or deletion? So in this case, if we sequence the area of genome containing this particular deletion or insertion, from the deletion or insertion side, two different templates appear, and we'll see the signals from both templates at the same time. So it is like here, and also insertion exactly the same. From the moment of insertion, we can see that two signals are appearing. And last question, what result will we get in Sanger sequencing if only 10% of cells in the analyzed tissue, tumor tissue for instance, have a given mutation? So let's go from left to right. Here we have 100% cells, they are all the same, and we have only one signal in particular position. Then we have the heterozygotes, so there are two signals, and we can see two peaks at the same position. And if there is 25%, 10%, 7%, or 0%, there is almost no difference, because if there is less than 50%, for instance 25, maybe we have some signal, but it's at the background level, and it's very difficult to distinguish the signal from this lower number of cells from the background. So Sanger sequencing method, in fact, cannot detect the mutation if it is in less percentage of cells than 50%.